So here are some things that will be in your microbiology toolbox, and especially in lab. These are things that we're going to see and cover. And so these are little cartoons, but I feel like they'll help kind of just get the idea across, and you'll see how they actually look in lab. So of course, we're going to need microscopes to be able to see these organisms that are microscopic. So microscopes, what they do, they produce magnified images of things that are too small to be seen by the naked eye. So to be able to see and observe cells like bacteria, we're going to need microscopes. We're also going to need stains and dyes because a lot of microbes don't have any color to them. They're like translucent. They don't need any color. They don't need to kind of like hide and blend into their environment. So a lot of times you need to add color to microbes to be able to see them and see their cells and different features. So we're going to add different stains and dyes that will give them a color that we can then help learn about them. So like if for a particular stain like the Gram stain, if they turn purple, that's going to help us see them, but that's also going to teach us something about the cell wall. So that's why we're going to add stains and dyes throughout the semester. They might teach us different things. Growth media is like stuff that helps us grow microorganisms in a lab setting. So growth media can be like a solid or a semi-solid. It could be liquid. It's kind of just something that you can grow microbes in or on. It typically has like stuff added that are like nutrients that are going to help the bacteria grow. Sometimes you can add different stains and dyes into the media. And so that'll help you see maybe color changes due to whatever the bacteria are producing. And so that's another thing that we're going to do a lot in lab. Grow them in different kind of media that is going to teach us more about what they do. So growth media is how we grow microorganisms in a lab setting. Petri dishes are used to help hold the growth media. So growth media, you can't just have like loose. You need a container for it because it might be either liquid or semi-solid. So Petri dishes are used to help hold growth media. So there is a kind of media called agar. And so agar is almost like Jell-O. So you mix it as a liquid and then you pour it and then it solidifies over time into like a little semi-solid. So Petri dishes are used to hold that kind of media, agar. And then when it solidifies, you can put the bacteria on top of the surface and see how it grows, in different ways. So like shown in this picture, there is growth media inside of this Petri dish. You can also have growth media that's liquid, and so for that, you might pour it into test tubes. So test tubes can be used to grow microbes in a liquid called a broth. You can also make it like a semi-solid or even just like a solid. Like you could still pour the agar in here and then let it solidify and then learn different stuff about it that way. So we're going to be using test tubes, and so test tubes are, glass and can break, and so that's part of the safety concerns that we're going to talk about in lab, is how to handle glassware responsibly and why you need to have closed-toed shoes because they can break, and if they have bacteria in them and you get a cut, boom, that just entered your blood. Test tubes are something we're going to use. Bunsen burners are also something we're going to be using, and so this little picture at the bottom, this is a Bunsen burner. It's like a way to produce a flame using gas, and the flame will be used to sterilize equipment. So for example, the next item is an inoculation loop. So it has like a handle, and then it's like a wire, and then the wire is long, and then at the very end there's a little loop. And so this is metal, and so what you can do is sterilize that in the flame. It kills off any microbes that might be there, and then it's sterile, and you can move around the microbes that you want to move around and not introduce any contamination. So these loops help either like streak microorganisms onto the growth media, or transfer from one kind of tube to another. They're our main tool to move the microbes around. But yeah, they're going to be metal so that we can sterilize them and reuse them in the lab. So again, we're going to have like a whole health or a lab safety talk. And so we're going to talk about all that in lab. But these are the tools that you can like expect to use. Okay, and then for the last section, we're going to talk a little bit more about microscopes. So microscopy, again, is necessary for microbes. And so you can read chapter 2, section 1 and 2, but they're kind of a lot of detail. And so I'm going to mainly focus on section 3 and 4. So what microscopes do, instead of just getting a magnifying glass or taking a picture, microscopes help increase the magnification of something, so of an object. It's the ability to enlarge the image of an object when compared to the real object. So say you're just looking at like a hair. That's looking at it, just by eye. And if you look at it under a microscope and it magnifies it, it might be magnifying it by 3 times. And so magnification is the ability of one certain lens to enlarge the image of an object. And then if you see something like 3x, 5x, that's telling you how much it magnifies something as compared to the real object. So there are different kinds of magnification. We'll talk about also in lab. So that's one thing that microscopes do. They magnify something, but they also have resolution. So resolution is the ability to tell 2 separate points or objects are separate. And so that might seem familiar when it comes to maybe your phone camera, or maybe even the resolution when you're watching a video. So resolution is, the ability to tell 2 separate points or objects are separate. And so in like a digital version, that's like pixels. Like there's tiny little dots that help make up an image that's digital. And so if there's higher resolution, there's more and more tiny dots, and it kind of looks better. And so when you use a microscope, it doesn't just magnify, it also increases the resolution. Like it looks clearer as you're looking up closer. So if you think about taking a picture on your phone and you're trying to see something in the distance, if you were to take a picture and then zoom in, you're increasing the magnification. You're kind of like zooming in on something, but that doesn't make the resolution better. It might still be fuzzy and not clear. So either you would use maybe a different like setting on your phone's camera that has better resolution. Sometimes phones now have multiple cameras to help you increase the resolution, or maybe you would just get closer. So if you wanted like an even better view of that thing, you would have to get closer, and that would help magnify and increase the resolution. But so that's what microscopes do. They both magnify something, but also increase resolution. Like you can really see in detail that image. Instead of it just being like blown up, but still crappy, it's also a better resolution. And so, those are the 2 main things that microscopes do. And then lastly, they also provide contrast. So there can be visible differences between parts of a specimen. So you can adjust the contrast. That way you see different like aspects of an image. So we'll talk more about this in lecture or in lab, but just to give you an overview of what microscopes do, why they're so important to the field. And so there are different types of microscopes. Simple microscopes have a single lens, so almost like a magnifying glass, or like the one that I showed you that Van Leeuwenhoek had. And compound microscopes have multiple lenses, so it's almost like having 2 magnifying glasses, and by working together, you magnify it even more. And so there are different types of compound microscopes. And so these are the 3 categories that we're going to talk about: light microscopy, electron microscopy, and then scanning probe microscopy. In lab, we're only really going to do light microscopy. That's all, we can really afford to be able to do. These kinds of microscopes are like super crazy expensive. And so we're not really going to get into that this semester, but light microscopy is what you're going to be doing in lab. So light microscopy uses lenses to focus light on a specimen to produce an image. And that's why it's a light microscopy. It's using different techniques to look at the image. And there are different kinds of light microscopes. So there is bright field microscopes, there are dark field microscopes, phase contrast microscopes, differential interference contrast microscopes, fluorescence microscopes, confocal microscopes, and two-photon microscopes. So each of them has their own benefits and why you would use that other one versus another one. And so we're mainly going to focus on bright field. I'm going to also talk a little bit in more detail about phase contrast and fluorescence, but just so you know, there's other ones too. So here's an example of a bright field microscope. It doesn't look exactly like the ones we're going to use in lab, but overall the parts are the same. So there is going to be an eyepiece, and these lenses are called the ocular lenses. So if you think of binoculars, they go closer to your eyes. The ocular lenses are going to be those that you look through. And so it's a combination between that lens, the ocular lenses, and these lenses down here that help you really magnify something. Again, this is a compound microscope, so it's one lens working with another lens. So these other lenses are called the objective lenses, and so those can actually be rotated to be different magnifications. So the ocular lenses provide one level of magnification, and then whichever objective lens you have in place provides another magnification. But these you can rotate out, so you can get a more far away view, and then a closer view, and then a closer view with the different objective lenses in place. So again, the ones in lab are going to look a little bit different, but that's still going to be a part that you see. And so to be able to rotate the objective lenses to be different, there's a revolving nose piece, and so you just turn it and it turns a different objective lens into place. Where you put your slide and your specimen is called the stage. Your specimen is the star and it wants to be on stage and have the light shine on it. And so it's going to basically be like a black square or rectangle. And then you put your slide here within these silver arms that help hold the specimen in place. And then you can move the stage around with these knobs. So these are XY knobs or mechanical stage knobs, and so they move the whole stage and thus move your sample and your slide left to right and then front to back. Or I guess I should do it like that. Left to right, and then front and back. So you can move the stage around by just moving these knobs instead of you having to move the stage yourself. Use these knobs. You can also move the whole stage up and down, and what that does is move it either closer or further to the lenses, and so that makes it either more clear or less clear based on your sample and where the stage is. So you can move this whole stage up and down by using these knobs here. This looks like just one black knob, but it's actually two. So there is this outer knob called the coarse focus knob. And so it moves it up and down, by a lot. You can visually see the stage move. And then the fine focus knob is the smaller knob, and that moves the same thing up and down, but just by a small increment, and so you can't see it visually moving, but it is moving the stage ever so slightly. And so by doing that, and you're moving the stage either further away from the lens or closer, you'll see as you look through the ocular lens, the image either get clearer or more blurry. And then there is a light source here. So either the illuminator or the light bulb basically is down here, and it shines the light through here through your sample and then through one lens into the other lens. And so there are other pieces right here that you can kind of fiddle with that will make the contrast change, maybe let more or less light through. And so those, we'll have to see what works for you, how you want to see your image. And then you can change the intensity of the light coming through. You can make it brighter or less bright. Yeah, so those are going to be the parts of the microscope. And so those you can only kind of get to a certain magnification with a bright field microscope. And then you would move on to other microscopes to get even closer. Because, yeah, the lenses can only do so much magnification. Another kind of lens and microscope is phase contrast. So phase contrast microscopes work a slightly different way. So here's a diagram of a phase contrast microscope. So here it shines light through a condenser lens. It condenses the light into one point. And then whatever you're looking at, your specimen, your sample, is going to either reflect the light or like refract it and bend it in a different way. And then it'll go through one lens, a phase plate, and then another lens that you see through. And so we're not going to do that in our lab, but you might, in your future career maybe. And this helps produce like a better contrast. So here is an example. So on the left is a bright field microscope image. So this is this cell. And you can see the cell, but on the right, using the phase contrast microscope, you can see more contrast between different parts of the cell. So you can see more detail on this cell. And this is actually a simple squamous epithelial cell, so like a skin cell basically. And so here you can see a little bit more of that dimension to it. And again, they are like almost invisible against the background. So that's why you would use a phase contrast microscope, or you would apply dyes so that you could see the cell a little bit better. You can also do fluorescence microscopy. So you can add dyes that are fluorescent and they will absorb in certain parts of the cell. So in this example, there are fluorescent dyes absorbed by these kinds of epithelial cells. And so they will emit different colors based on like what structure they're bound to or what dye it is. So different cell structures absorb different dyes. Nuclei generally are stained blue. And so there's this stain called DAPI. It stands for all this. And so yeah, typically nuclei get stained blue with DAPI. And then other parts of the cells will be stained with different dyes that will mark that particular part of the cell. So there are structures called microtubules, and those are going to be marked in green. And then other like actin filaments are going to be labeled with red. And so that's how you can learn even more about like the cell biology of that particular kind of cell. So that would be fluorescence microscopy. And then another way of seeing fluorescence is through a different technique called immunofluorescence. And so in this case, it's usually like something that is fluorescent, some kind of fluorescent stain that's marks antibodies. And the antibodies will bind to something and show you something. So here's an example of an immunofluorescent stain to help visualize Neisseria gonorrhea, the bacteria that causes gonorrhea. Again, this should be italics, my bad. And so if the antibody that's marked with a certain dye or stain finds gonorrhea, it'll mark them, and then that's why you could see them now, because they're marked with the immunofluorescent tag. And so you can do that in different ways. You can tag like the thing you're looking for directly, or you can tag an antibody that normally tags that kind of thing. Yeah, so those are different forms of immunofluorescence. It's like using antibodies with like an attached fluorescent dye to see something. And so this is the overall picture summary of different kinds of microscopes, but again, kind of the major one that we're concerned about for this class is like bright field, and fluorescence. And then maybe phase contrast. But again, we're only going to use bright field in our labs. So another overall big category, so those again were light microscopes, light microscopy. So another huge category. Oh, and then notice also light microscopes can magnify up to about 1,000 times. And they are light microscopes because they use visible or UV light to produce an image. Electron microscopy is different. It works in a little bit of a different way, or a major different way. So electron microscopy focuses electrons on the specimen using magnets. And so this microscopy produces much greater magnification than light microscopy. It can magnify either 20 to 100,000 times or more one image. And there are two basic types of electron microscopy: transmission electron microscopy and scanning electron microscopy. And so, yeah, we won't really use those here, but just so you know, like this is how you get even closer and focus and study parts of cells even closer. So here's how you can see, for example, viruses. Viruses are so tiny, you need a whole different kind of microscope to be able to see them. And then even closer are scanning probe microscopes. So these produce images of even greater magnification by measuring feedback from sharp probes that interact with the specimen. Like there's something like probes that kind of like stab samples, and help show that. And so probe microscopes include scanning, tunneling microscopes, and atomic force microscopes. And again, that's way beyond our scope, but just so you know. So these can magnify from 100 to 100 million times or more. And they use very short probes that are passed over the surface of the specimen and like actually touch it and interact with it and produce an image. But yeah, that's tiny, tiny stuff. That's usually more for like materials science. Okay. So, like I mentioned, to be able to actually see microbes, you usually have to like treat them with stains or dyes. So most cells or microorganisms that we observe under a microscope lack any color or contrast, and so we have to add stains or dyes to be able to view them. And to be able to add stains or dyes and not have it just wash all over the microscope, you have to kind of properly prepare the specimens and samples so that you can do all this stuff and then do microscopy. So this may involve fixation. So you're fixing the cells to this microscope slide so they don't like come off. And then you could do staining. And then if you're doing other kinds of microscopy, you might need to do, other stuff to your sample. But that's like the main thing that we're going to be doing in lab to be able to see our bacteria. So you fix the bacteria onto the microscope slide, usually just passing it over the flame. Like with the heat, this kind of makes the cells like stick onto the slide. And then you could do different staining techniques depending on what you want to learn about the cells. So there are a variety of staining techniques that can be used with light microscopy, including gram staining, acid-fast staining, capsule staining, endospore staining, and flagella staining. And so these are all things that we're going to do in lab. And so I would still recommend reading this section of the textbook because it is going to be relevant to you. It's something you're going to be doing, even though we're going to do it more in lab. So these are all staining techniques that we're going to be doing, I think, except the flagella staining. And they all teach you something different about the cell. So it's not just to be able to see the cells, it's also teaching you about something. So these cells are Bacillus anthracis. Again, this needs to be italics. So they absorb a specific stain called crystal violet. And so there are some stains that are called positive stains because they stain the cell. There are other stains that stain the background. And so in doing that, if it stains everything except the cell, you can kind of see the cell a little bit better actually than if you did a positive stain. And so here in part C, this is a negative stain. It's kind of like producing like a photograph negative. It stains everything else, and then the actual thing is clear, the actual cells that you're trying to see. And there are pros and cons of each, you know. Yeah. And so here's another example. So the gram stain will stain some cells purple based on their cell wall composition, and then they'll stain other cells pink. And so we'll again talk more about this in lab, but that's how you learn more about cell types, like what they look like, but also their composition of their cell wall. So a main one is the gram stain.